Thank you very much, Natasha, for organizing this meeting and for giving me the opportunity to present. So our jumping off point is this idea that we're in brain imaging, so most of the time we make brain maps. And these brain maps are, if you're in psychology, you've designed a clever task and you find a pattern of activation. If you're in some clinical field, you've contrasted patients and controls for cortical thickness or something like this, and you have a brain map. And your next question is, obviously, what does this brain map look like? Does it co-localize with any canonical features of brain structure and function? The reason you want to know this, well, first of all, what I mean by that is things like gene expression, metabolic markers, different layers, neurotransmitter receptors, and so on. The reason you want to know this is, A, you want to understand something about the underlying mechanism, what gave rise to this pattern that you're seeing. And B, you want to know what the next set of steps are going to be for your follow-up experiments. And it's not as though this data is completely inaccessible to us. We can get a lot of this through garden variety MR scans. We can get it with other more exotic protocols. We can get it from other technologies that are maybe not available in our centers, but that are available through open science data sharing efforts across the globe. And so what I've chosen to do today is tell you a little bit about a toolbox that we've developed. It's kind of a nice little microcosm of all the challenges that this very simple but fundamental problem entails. And the toolbox is called NeuroMaps. It's kind of like a Google Maps for the brain. And the idea is basically this. It kind of has three components to it, which you don't have to use in sequence. You don't have to use all of them. But the idea is you bring in a map of your choice, this map that you've created, that you spent your PhD working on, and you would first provide you with a suite of transformations so you can easily go back and forth between the major coordinate systems that we have in brain imaging, something that I think a lot of trainees will appreciate. We provide a curated library of various features of brain structure and function that you can make comparisons against. And then finally, we provide a suite of statistical models that allow you to make these comparisons in a way that circumvents a lot of the problems that come with smooth brain maps, which I'll talk about as well. And then at the end of the day, if you really want to go through all of this, you'll get kind of an enrichment score, kind of like what you get with any bioinformatics tool. So I'll kind of take you through each of these. The library itself goes a little bit like this. Basically, we have a whole bunch of maps that have to do with microstructure, so both from PET and MRI, things like intracortical myelin from T1, T2, also synapse density that comes from the famous UCBJ PET tracer. We have various maps of metabolism, kind of looking at metabolism from different angles, again, using both MR and PET. We have maps of different kind of evolutionary and developmental expansions. So these are maps of how much the cortex changes over phylogeny and ontogeny, relative, in both cases, relative to non-human primates. Using source-resolved MEG as well as intracranial EEG, we give you maps of power, spectral power in the different electrophysiological frequency bands, the canonical frequency bands, as well as things like intrinsic time scale, a bunch of others. Working with the histological big brain atlas, so this is something that was developed by Alan Evans and company. It's a 20 micron Merkur stained atlas. So these are maps of cell bodies and how many cell bodies there are in the canonical 6 layers of the cortex. And using data from gene expression, so this is from the Allen Human Brain Atlas, and applying cell type deconvolution, we have maps of various cell types across the brain. Now, if you'd asked me when we started this, whether, what is my favorite annotation, what is it that I want, what's on my wish list, I wouldn't have said any of the ones that I just showed you. What I think most of us are going to be interested in by default are neurotransmitter receptors. And the reason for that is that we know that neurotransmitter receptors modulate the link between brain structure and function, and they remain to this day, our main way of kind of interfacing with the brain, of modulating the brain pharmacologically. So we can get information on different neurotransmitter receptors through PET. The only problem is that oftentimes these data sets are collected in isolation by different groups. So we went and asked a few years ago bunch of people first locally at the pet center at the MNI, if they do pretty please be willing to share data with us. And then they did, and then we asked some more people, their friends, and then they asked their friends. And so at the end of the day, we had something that looked like this, which is not that exciting, but what it is really a set of brain maps. It's a chemoarchitectural whole brain atlas of the human brain. So it has maps for more than, at this point, more than 20, exactly 20 receptors and transporters across 9 different neurotransmitter systems. That's all available in the toolbox. We also have a contribution pipeline, so when people have a cool map that they want to send us, we entertain that possibility. I mentioned briefly that we had some data on gene expression. We actually have another tool called Avogen that we developed for specifically working with gene expression data. So the tool will, with one line of code, access data from the Allen Human Brain Atlas. They'll download it and pre-process it for you. It typically takes about a minute. And then what you can do with this, I mean, the possibilities are endless, but you can do all the stuff you might have seen in the literature, like do region of interest analysis. You can mask out a region. You can focus on specific genes that you're interested in, ApoE or SNCA, things like this. It'll work with any atlas that you give it, and you can also do dense interpolation. And the last thing I want to say is it's not going to cook your dinner for you, but it will write your method section for you. So depending on the series of pre-processing options that you pick, it'll actually spit out a manuscript-ready method section that you can put in. And this is not like to be cheeky or anything, but the point was that we started to realize that a lot of people, even though they were seemingly working with the same data, occasionally would get slightly different results, and it wasn't always clear why this was happening. And so this at least is a step in the direction of standardizing workflows in the field. So the point being that you don't have to get summaries of different features of gene expression. You can get any gene expression map that you like as well. Okay. We worked very hard to provide these maps in their native space. So we didn't, whoever collected them, we provide them in the space that they made them available in. What this means is that now we have this problem of different groups using different spaces. And so we have a suite of transformations. We provide support for the major coordinate systems in brain imaging. So MNI 152, CIVET, FS average, and FSLR. And if you want to go from volume to surface, we've implemented registration fusion. If you want to go between the different surface spaces, we've implemented multimodal surface matching, or MSM. We didn't invent either of these two methods, but we extensively tested them and benchmarked them. And at least in our hands, they are really as good as advertised. And what we find is that in day-to-day life, this is what trainees use most. And we also have a function for parcellating. So if you bring in any atlas that you like, you can get your parcellations as well. Okay, so we have the library, we have the transformations. So you can take your map, you can bring it into the space of your target map, or vice versa. You want to compare them, and this is where the fun starts, because anytime you want to engage in this business of quote-unquote contextualizing brain maps, you're gonna at some point attempt to correlate your two brain maps. And the problem is that our brain maps entail a lot of spatial autocorrelation. What that means is that areas that are close together tend to be similar, areas that are far apart tend to be different. Some of this is purely, biological reality. Another aspect of this is that we tend to smooth all of our maps in virtually any brain imaging processing pipeline. And I just want to tell you why this is a problem. The problem is you're going to get inflated p-values. And so here what I've done is generated two brain maps that are completely random. They should not be correlated with each other, and they're not. What I'm going to do now is apply just a simple filter, a smoothing filter. I'm going to start to get maps that look a lot like what we see in various imaging modalities. And when you do that, you start to get very, very, very high correlations that should not be there because the maps were random to begin with. So what's happening is that when you have greater spatial autocorrelation, those, if you think about, plotting a scatter plot of brain map 1 versus brain map 2, the points there, the brain regions are not independent. You're losing your true degrees of freedom, and so you're getting spurious correlations. We at the time that we kind of started to do this, we realized that there were actually many ways in the field of dealing with this, famous spin tests and other things that people might have heard of. We've implemented all of them. So these are just, thank you. These are just different ways of controlling for spatial autocorrelation when you run correlations between brain maps. We've implemented all of them. We've benchmarked all of them. You can have the reference there where we kind of lay out exactly when and which method should be used, but all of them are in Neuromaps and ready to be used. And so that's kind of my plug for Neuromaps. And I just want to give you for the last 5 minutes the reason why we did this and where I think it's leading us. So one example of how this can be used, just one example, is we pulled data from the Enigma Consortium, which, as many people know, this is a kind of big data sharing group that deals mainly in cortical thickness maps for various diseases. So at the time that we did this, we found data on 13 different diseases that are shown there, and the kind of patterns of cortical thinning. So these are case control contrasts, and the maps are cortical thinning. And what we asked was very simply, using Neuromaps, whether these patterns of cortical thinning in these different diseases correspond to patterns of neurotransmitter receptors and transporters. And so that's what you're seeing here. The horizontal bars are telling you how well I can predict a pattern of cortical thinning in a disease using maps of neurotransmitter receptors. So you can see that actually the results are really good across the board, except for, it's very small, but except for idiopathic epilepsy, where I'm not really sure that you would even expect a neurotransmitter signature, and schizotypy, which is not really a disease, more of a trait. And then what you have here now is a really cool lookup table that tells you, what are the levers you should be pressing if you wanted to modify this particular phenotype. So it's a nice mixture of things that you expect based on prior knowledge. The textbook examples, like if you see the serotonin transporter there being kind of the top hit for a lot of the psychiatric diseases, but then oftentimes you see evidence of hits that maybe you don't necessarily think of as being the main receptor that's implicated in disease, but there is support for it in the literature, like the histamine 3 receptor in Parkinson's. Okay, and the reason why we were really into this is that we wanted to know how a lot of these biological features relate to brain wiring. So our lab is called the Network Neuroscience Lab, which means that we're really obsessed with how areas are connected with each other. What you can do is you can take these maps of annotations and you can superimpose them on our models of the connectome, so wiring diagrams that we get from diffusion-weighted MRI or resting state fMRI. You can mix and match, exactly see exactly which brain areas should be endowed with what value of from a particular map. And you get these beautiful objects that we call annotated connectomes that basically tell you both who's connected to whom, but then also what are their local features. And so we can generate annotated connectomes basically for all of the things that we just talked about, different features of dynamics, myelin, metabolism, and so on. And what that does for us is that it allows us to ask now newer questions that get us a little bit closer to the biology. So we work a lot on models of disease spread, this idea that in many neurodegenerative syndromes, you have misfolded proteins that move transsynaptically, but their movement is also guided by local vulnerability of different neuronal populations. So you can use information about cellular composition to make those models better. If we kind of go with this notion that there are a lot of spatial variations in microarchitecture, we can also ask, well, what are the dynamical consequences of that? So we look at how different features of dynamics co-vary with microarchitecture. We've also used these biologically enhanced models to make predictions about where you should apply different types of brain stimulation to get desired cognitive effects. And finally, we've also done a lot of work looking at how you can take biologically derived connection patterns, implement those as artificial neural networks, and ask questions about, if we have inter-individual differences in brain connectivity or other local biological features, what are the cognitive and computational consequences of that? So I don't have any concluding slide except to say that, the message that I was going for is really simple, that if you pay attention to the biology, I think you can make more biologically realistic models, and that the data and the methods are already here, and so it's an exciting time. Thanks for your attention.